Hi, it's Miss Parrott, and welcome back to the AP Bio Exam Skills Series. In this series, we break down not just the content, but strategies that you can use to be successful on the day of the AP Bio exam. So, one of the biggest problems with the AP Bio multiple choice section, it's not that you know nothing and that you have no idea. It's that when you get down to two strong answers, which one do you choose? Today we're going to look at a repeatable strategy, a work example, and common mistakes to avoid. So stick around to the very end. Okay, so here's our strategy. Step one. Step one, reread the question stem. Now, the question stem is a part of the question with an actual question mark. It's not like all the background and all that deep stuff. You've already done that once, right? You're down to two possible answers. We're going to reread the question stem and think about what is the question actually asking, right? Slow down first. Like, I need to slow down on this video. Slow down. Identify your task verbs. Is it asking you to predict, to justify, to explain? These are all different things, and AP Bio likes to give you a right answer, but not to the question that's being asked. So identify your task verb and choose that. Wrong answers will often be, again, true, but answer a different question. Or they might give you, like, if the question says, describe and explain why. Well, it might give you two things that correctly describe, but the explain why, only one of them is the correct explanation. That's the one you'll choose. So, after you've reread the question, if you still have two strong answers, identify the core concept. Ask yourself, what AP Bio concept is being tested here? Is it enzyme specificity, gene expression, energy transfer? Sometimes AP Bio will give you a partially right answer in a context that has nothing to do with the rest of what's happening. So the correct answer will match the concept that's being asked about precisely, whereas an incorrect answer might have the right biology, but like in a totally wrong context or topic. It may sound super sciencey, like they'll use lots of big words, but again, it doesn't match the concept. So if the concept is energy transfer and it's asking you stuff about, like, it's giving you a context of gene expression, that's not going to be right. And sometimes they just give you stuff that's irrelevant to the topic. So we may be like, there was a good example of one where it was an osmosis question, right? And it's asking about water concentration. And yes, that involves membranes, but one of the answer choices had something about sodium potassium ion pumps, which, yeah, those are proteins in membranes, but that has to do with cell signaling in the nervous system, not osmosis. So we choose the osmosis answer. All right. What if we have still two good answers and we can't figure it out? Step 3 is to match the answers to the data. Lots of questions. And again, this is a pro because it means you can figure it out even if you don't know what it's talking about. But it's a con because sometimes you get freaked out if you don't know what something's talking about. So if they give you an experiment, a data table, a graph, ask yourself, does the data, not just like my general knowledge of the topic, but does the data given either on the graph in the picture or in the data table, does that support my answer? The correct answers, they'll not go beyond the data. The data will give you the answer. It will be there. You can see it. You can point to it in the data table or explain it on the graph. Wrong answers often require assumptions that are not explicitly given in the experiment. They may be generally true, but not match the data in the actual experiment that the exam describes. Or they may be vague and imprecise. Precision is a really important part, like the answers matching the question precisely. So precision is important. So if it's too vague, it's probably not the right answer. Go with the more precise one. All right, let's look at an example together. And so this is one that's already been narrowed down to 2 pretty good answers. All right, so we have a scientist increases the temperature of an enzyme catalyzed reaction from 20 degrees C to 40 degrees C. The rate of reaction increases. Which explanation supports this observation? So this may come with a data table or a graph or not, because all the data is in the information. So, but if let me grab a little marker here. Imagine it's saying that it went from 20 to 40, and that's the temperature, right? And the rate of reaction, it says it increased. Cool. So we've gotten it down to 2 answers. Higher temperature increases molecular motion, leading to more frequent enzyme substrate collisions. Sounds pretty good. Let's see what the next one is. Higher temperature increases enzyme concentration, which increases reaction rate. Okay, so let's think through our steps, right? First, reread the question stem. Which explanation supports this observation? So the observation that we're seeing an increase in rate. Awesome. Then, so these are both explanations. So it's not like one of them is like predicting or justifying when we should be explaining. So we did reread, but we still have 2 pretty okay answers. All right, our second step, what's the concept? Well, our concept is enzyme substrate, like impacts of enzymes on reaction rate. In this case, in the context of temperature. So higher temperature increases molecular motion. Yeah, right? That's like it increases kinetic energy, right? Leading to more bump into each other, more enzyme substrate collisions. So you're going to get a higher rate. So far, feeling good. This one, higher temperature increases enzyme concentration. So let's pause there, because higher temperature, it does make things move faster, but it doesn't add more enzyme. Increasing enzyme concentration means there's more enzyme than like in the space. So let's think about question 3, which says look at the data. I mean, technically, like say you were in a water environment and the temperature got higher and then some of the water evaporated off and then the enzyme concentration increased. Maybe, but again, it's requiring that I make assumptions that are not described in the question. So there's no indication that higher temperature itself doesn't increase enzyme concentration. Now, again, you could do like I just did and chase it down a rabbit hole and get there, but this is not really supported by the data in the question. And 40 degrees, it would take a while at 40 degrees to really significantly change enzyme concentration. So yeah, higher temperature increases reaction rate, but it doesn't necessarily increase enzyme concentration. All right, so we're going to choose A because all parts of this are true. They match the data described. We don't have to make outside assumptions. It answers the question precisely, so we're all good. Higher temperature does increase molecular motion, leading to more frequent enzyme substrate collisions, which is when enzyme and substrate come in contact. And if they come in contact, enzyme's going to do its action, and then the products will be produced. All right, so a quick rundown again of things to avoid and tiebreakers. So tiebreakers to ask yourself when you're down to your two things: what's my task verb? What is the concept that is truly asking about? What did the data say? Is my answer actually relevant to the situation? Is it precise? And consider all your variables, right? Is it the measured? Is it the manipulated? Is it a control? You want to avoid extreme wording like always and never. And that's a little relationship advice too. But avoid extreme wording, avoid vague answers, avoid anything that's going to require ungiven assumptions, question or answers that are overly wordy just for the sake of being overly wordy. Ignore those. And you got to pay attention to your graphs and data. Don't ignore. If they spent the time to print out the graph and the data, it's probably useful to you. Thinking like a test writer is what's going to help you go from I understand how photosynthesis works to I can answer questions about photosynthesis on the AP Bio exam quickly and efficiently. And that's our goal here on the AP Bio exam skills series. If you like this video, please do give it a thumbs up. For more in this series, subscribe to my channel. Leave a comment, a question, or an emoji down below. I'll get back to you for sure. Teachers, TPT resources that go with this video and all the rest of my AP exam skills series will be coming soon. There's already a whole bunch of resources that have to do with my AP Bio Equations Explained series where I break down all the equations and formula sheet. So check that out. Keep knowing, keep learning, keep growing, and we'll see you next time.